DNA replication. Here we have a little DNA strand, and its top strand is a leading strand because it's read 5' to 3'. DNA is always read 5 to 3. And the bottom of this little DNA strand is called the lagging strand because it's the one that's read, or well, it's read 5' to 3', but it starts with 3', ends with 5'. Now, probably wondering what a little DNA strand is. DNA is the genetic code that basically makes up you, and it's made up of these little nucleotides. This first little nucleotide is guanine, and it always connects to cytosine. And then we have adenine, and that connects to thymine. This little white ball here, it's helicase. Helicase is going to come over to our DNA strand and unzip it. Oh, there goes helicase. And these little things are called single-strand binding proteins. And what their job is as helicase is unzipping our DNA, so they go in and fit in between the nucleotides, but don't cover up the nucleotides, to keep our DNA strand from snapping back into its double helix form, so that DNA replication can happen, which we are going to see pretty soon. There we go. Our little single-strand binding proteins fitting in those nucleotides. Helicase unzipping. Yeah. There we go. Almost done. Okay. And now, this little thing. That is RNA primase. What that does is it goes and primes the first little part of our DNA strand, just, to give it a little startup, because then this little thing called DNA polymerase 3 is going to come in and finish the job. One thing you want to know about RNA primase is that since it's RNA, it doesn't have thymine. Instead, it has this little thing called uracil, and that just binds to the adenine instead of thymine. Alright, there goes our little RNA primase, and here comes the DNA polymerase, finishing up the strand, the leading strand that went 5 to 3. Now we gotta start over here on the end of the lagging strand. Since it starts with 3' and ends with 5', we have to start at that little 5' end where the helicase is finished up. RNA primase is gonna come in and start us off, and then we have to have DNA polymerase finish up that a little section, but then the RNA primase is going to have to come in again and prime, and then the DNA polymerase is going to go again, and it keeps starting over and over and over. That's why this little thing is called the lagging strand, because it lags behind. Alright, we call these segments that the lagging strand is read in Okazaki fragments. Now this is DNA polymerase 1. Before we had just seen DNA polymerase 3. But what 1 does is it comes in and finds those spots where the RNA primase had started off, and it finds those uracils and replaces them with thymine, so that we have DNA, not some half DNA, half RNA thing. Okay, now this yellow thing is called ligase, and what it does is it comes along our new DNA strand and it makes it a, cemented DNA strand. It just has little magical powers, and it makes it so it can be on its own and gets rid of those little single strand binding proteins, and then we have 2 DNA strands. Hooray!